Abstract. Background. Severe acute respiratory syndrome coronavirus 2, SARS-CoV-2, infection and the resulting coronavirus disease 2019, Covid-19, have afflicted tens of millions of people in a worldwide pandemic. Safe and effective vaccines are needed urgently. Methods. In an ongoing multinational, placebo-controlled, observer-blinded, pivotal efficacy trial, we randomly assigned persons 16 years of age or older in a 1-to-1 ratio to receive 2 doses, 21 days apart, of either placebo or the BNT162b2 vaccine candidate, 30 per dose. BNT162b2 is a lipid nanoparticle formulated, nucleoside-modified RNA vaccine that encodes a perfusion-stabilized, membrane-anchored SARS-CoV-2 full-length spike protein. The primary endpoints were efficacy of the vaccine against laboratory-confirmed COVID-19 and safety. View full size. Results. A total of 43,548 participants underwent randomization, of whom 43,448 received injections. 21,720 with BNT162b2 and 21,728 with placebo. There were 8 cases of COVID-19 with onset at least 7 days after the second dose among participants assigned to receive BNT162b2 and 162 cases among those assigned to placebo. BNT162b2 was 95% effective in preventing COVID-19. 95% credible interval, 90.3 to 97.6. Similar vaccine efficacy, generally 90 to 100%, was observed across subgroups defined by age, sex, race, ethnicity, baseline body mass index, and the presence of coexisting conditions. Among 10 cases of severe COVID-19 with onset after the first dose, 9 occurred in placebo recipients and 1 in a BNT162b2 recipient. The safety profile of BNT162b2 was characterized by short-term, mild to moderate pain at the injection site, fatigue, and headache. The incidence of serious adverse events was low and was similar in the vaccine and placebo groups. Conclusions. A 2-dose regimen of BNT162b2 conferred 95% protection against COVID-19 in persons 16 years of age or older. Safety over a median of 2 months was similar to that of other viral vaccines, funded by BioNTech and Pfizer. Clinical Trials Gov number, NCT 04368728 opens a new tab. Digital object thumbnail quick take safety and efficacy of the BNT162b2 COVID-19 vaccine 3 o'clock coronavirus disease 2019, COVID-19, has affected tens of millions of people globally since it was declared a pandemic by the World Health Organization on the 11th of March, 2020, 2 older adults, persons with certain coexisting conditions, and front-line workers are at highest risk for COVID-19 and its complications. Recent data show increasing rates of severe acute respiratory syndrome coronavirus 2, SARS-CoV-2, infection and COVID-19 in other populations, including younger adults. 3 safe and effective prophylactic vaccines are urgently needed to contain the pandemic, which has had devastating medical, economic, and social consequences. We previously reported Phase 1 safety and immunogenicity results from clinical trials of the vaccine candidate BNT162b2. 4 a lipid nanoparticle formulated, 5 nucleoside-modified RNA, modRNA, 6 encoding the SARS-CoV-2 full-length spike, modified by 2 proline mutations to lock it in the perfusion conformation. 7 findings from studies conducted in the United States and Germany among healthy men and women showed that 230 doses of BNT162b2 elicited high SARS-CoV-2 neutralizing antibody titers and robust antigen-specific CD8+ and 1-type CD4+ T cell responses. 8 the 50% neutralizing geometric mean titers elicited by 30 of BNT162b2 in older and younger adults exceeded the geometric mean titer measured in a human convalescent serum panel, despite a lower neutralizing response in older adults than in younger adults. In addition, the reactogenicity profile of BNT162b2 represented mainly short-term local, that is, injection site, and systemic responses. These findings supported progression of the BNT162b2 vaccine candidate into Phase 3. Here, we report safety and efficacy findings from the Phase 2/3 part of a global Phase 1, 2, 3 trial evaluating the safety, immunogenicity, and efficacy of 30 of BNT162b2 in preventing COVID-19 in persons 16 years of age or older. This data set and these trial results are the basis for an application for emergency use authorization. 9 Collection of phase 2/3 data on vaccine immunogenicity and the durability of the immune response to immunization is ongoing, and those data are not reported here. Methods, trial objectives, participants and oversight. We assessed the safety and efficacy of 230 doses of BNT162b2, administered intramuscularly 21 days apart, as compared with placebo. Adults 16 years of age or older who were healthy or had stable chronic medical conditions, including but not limited to human immunodeficiency virus, HIV, hepatitis B virus, or hepatitis C virus infection, were eligible for participation in the trial. Key exclusion criteria included a medical history of COVID-19, treatment with immunosuppressive therapy, or diagnosis with an immunocompromising condition. Pfizer was responsible for the design and conduct of the trial, data collection, data analysis, data interpretation, and the writing of the manuscript. BioNTech was the sponsor of the trial, manufactured the BNT162b2 clinical trial material, and contributed to the interpretation of the data and the writing of the manuscript. All the trial data were available to all the authors, who vouch for its accuracy and completeness and for adherence of the trial to the protocol, which is available with the full text of this article at NEJM.org. An independent data and safety monitoring board reviewed efficacy and unblinded safety data, trial procedures. With the use of an interactive web-based system, participants in the trial were randomly assigned in a 1-to-1 ratio to receive 30 of BNT162b2, 0.3 ml volume per dose, or saline placebo. Participants received 2 injections, 21 days apart, of either BNT162b2 or placebo, delivered in the deltoid muscle. Site staff who were responsible for safety evaluation and were unaware of group assignments observed participants for 30 minutes after vaccination for any acute reactions. Safety. The primary endpoints of this trial were solicited. Specific local or systemic adverse events and use of antipyretic or pain medication within 7 days after the receipt of each dose of vaccine or placebo, as prompted by and recorded in an electronic diary in a subset of participants. The reactogenicity subset, and unsolicited adverse events, those reported by the participants without prompts from the electronic diary. Through 1 month after the second dose and unsolicited serious adverse events through 6 months after the second dose. Adverse event data through approximately 14 weeks after the second dose are included in this report. In this report, safety data are reported for all participants who provided informed consent and received at least 1 dose of vaccine or placebo. Per protocol, safety results for participants infected with HIV, 196 patients, will be analyzed separately and are not included here. During the phase 2/3 portion of the study, a stopping rule for the theoretical concern of vaccine enhanced disease was to be triggered if the one-sided probability of observing the same or a more unfavorable adverse severe case split, a split with a greater proportion of severe cases in vaccine recipients, was 5% or less. Given the same true incidence for vaccine and placebo recipients, alert criteria were to be triggered if this probability was less than 11%. Efficacy. The first primary endpoint was the efficacy of BNT162b2 against confirmed COVID-19 with onset at least 7 days after the second dose in participants who had been without serologic or virologic evidence of SARS-CoV-2 infection up to 7 days after the second dose. The second primary endpoint was efficacy in participants with and participants without evidence of prior infection. Confirmed COVID-19 was defined according to the Food and Drug Administration, FDA, criteria as the presence of at least one of the following symptoms: fever, new or increased cough, new or increased shortness of breath, chills, new or increased muscle pain, new loss of taste or smell, sore throat, diarrhea, or vomiting, combined with the respiratory specimen obtained during the symptomatic period or within 4 days before or after it that was positive for SARS-CoV-2 by nucleic acid amplification-based testing, either at the central laboratories or at a local testing facility, using a protocol-defined acceptable test. Major secondary endpoints included the efficacy of BNT162b2 against severe COVID-19. Severe COVID-19 is defined by the FDA as confirmed COVID-19 with one of the following additional features: clinical signs at rest that are indicative of severe systemic illness, respiratory failure, evidence of shock, significant acute renal, hepatic, or neurologic dysfunction, admission to an intensive care unit, or death. Details are provided in the protocol. An explanation of the various denominator values for use in assessing the results of the trial is provided in Table S1 in the supplementary appendix, available at nejm.org. In brief, the safety population includes persons 16 years of age or older. A total of 43,448 participants constituted the population of enrolled persons injected with the vaccine or placebo. The main safety subset as defined by the FDA, with a median of 2 months of follow-up as of the 9th of October, 2020, consisted of 37,706 persons, and the reactogenicity subset consisted of 8,183 persons. The modified intention to treat, MIT, efficacy population includes all age groups 12 years of age or older, 43,355 persons. 100 participants who were 12 to 15 years of age contributed to person time years but included no cases. The number of persons who could be evaluated for efficacy 7 days after the second dose and who had no evidence of prior infection was 36,523. And the number of persons who could be evaluated 7 days after the second dose with or without evidence of prior infection was 40,137. Statistical analysis. The safety analyses included all participants who received at least one dose of BNT162b2 or placebo. The findings are descriptive in nature and not based on formal statistical hypothesis testing. Safety analyses are presented as counts, percentages, and associated Klopper-Pearson 95% confidence intervals for local reactions, systemic events, and any adverse events after vaccination. According to terms in the Medical Dictionary for Regulatory Activities, MedDRA, version 23.1, for each vaccine group. Analysis of the first primary efficacy endpoint included participants who received the vaccine or placebo as randomly assigned, had no evidence of infection within 7 days after the second dose, and had no major protocol deviations. The population that could be evaluated. Vaccine efficacy was estimated by 100, 1I, where I is the calculated ratio of confirmed cases of COVID-19 illness per 1,000 person-years of follow-up in the active vaccine group to the corresponding illness rate in the placebo group. The 95.0% credible interval for vaccine efficacy and the probability of vaccine efficacy greater than 30% were calculated with the use of a Bayesian beta binomial model. The final analysis uses a success boundary of 98.6% for probability of vaccine efficacy greater than 30% to compensate for the interim analysis and to control the overall type 1 error rate at 2.5%. Moreover, primary and secondary efficacy endpoints are evaluated sequentially to control the family-wise type 1 error rate at 2.5%. Descriptive analyses, estimates of vaccine efficacy and 95% confidence intervals, are provided for key subgroups. Results. Participants. Figure 1. Enrollment and randomization. Table 1. Demographic characteristics of the participants in the main safety population, between the 27th of July, 2020, and the 14th of November, 2020, a total of 44,820 persons were screened, and 548 persons 16 years of age or older underwent randomization at 152 sites worldwide. United States, 130 sites, Argentina, 1, Brazil, 2, South Africa, 4, Germany, 6, and Turkey, 9, in the Phase 2/3 portion of the trial. A total of 43,448 participants received injections. 21,720 received BNT162b2 and 21,728 received placebo. Figure 1. At the data cutoff date of the 9th of October, a total of 37,706 participants had a median of at least 2 months of safety data available after the second dose and contributed to the main safety data set. Among these 37,706 participants, 49% were female, 83% were white, 9% were black or African.